As you know, this is a very dire indication. Immunotherapy has really changed the way in which we treat patients with melanoma, but there are many patients that are resistant to this treatment.
All right, welcome back inside our Proactive newsroom. And joining me now is Dr. Francesca Barone. She is the Chief Scientific Officer for Candel Therapeutics. And Dr. Barone, great to see you. How are you?
I'm good, very well, in a beautiful day in Houston here in Texas. It's great excitement for SITC.
Good stuff. Well, I'm glad to have you along. I'm very excited about the news that you had out from the company talking about your drug CAN-3110. Before we go any further about what you'll be presenting at a conference, maybe just remind everyone a little bit about that.
Yeah, so CAN-3110, as you say, CAN-3110, we call it CAN-3110, is a first-in-class oncolytic virus. It's different from all the other viruses that have been developed in the herpes space because whilst all the other viruses have a deletion of a specific gene that support replication, but sometimes it's also linked to neurovirulence, what we've done here is that we've engineered this virus to maintain the capability of replicating specifically in cancer cells because its replication is under a specific promoter that is expressed by cancer cells. So it spares normal tissue while it replicates and kills cancer cells. And whilst doing that, it also elicits a very strong immune response against the cancer cells. And we have very compelling data in recurrent high-grade glioma, where this study is in a phase 1b/2a clinical trial.
Yeah. What uses would you have for this? You mentioned cancer, obviously, so that's the significant one, but it also has potentially other ones as well?
Yeah, well, the virus was specifically developed at the beginning for recurrent high-grade glioma because of the expression of nestin that regulates the promoter, so the capability of replicating these cells. But actually, what we are presenting here is the fact that we can expand the use of this virus beyond high-grade glioma. And so what we are testing is other tumors that are characterized by the expression of nestin, and there are several. And we are testing how this virus can actually be used in these other indications.
Yeah, and you're going to be presenting some of this data at a conference coming up in Houston. And in fact, that's one of the other uses that you're talking about. You're going to be presenting the data on, correct?
Correct. So what we're doing here, we've put together a package, a preclinical package for the use of CAN-3110 in melanoma. As you know, this is a very dire indication. Immunotherapy has really changed the way in which we treat patients with melanoma, but there are many patients that are resistant to this treatment. And so we are presenting data that show that CAN-3110, when injected in the tumor of the mice, so first of all, in vitro, we have evidence that they kill melanoma cell lines. We have a couple of human cell lines of melanoma. And also, when we put this in tumor models of melanoma in vivo, we have a significant reduction of tumor burden and a significant activation of the immune system. These are data that are very similar to what we've already observed in humans with this agent in recurrent high-grade glioma. And as you can see, melanoma and high-grade glioma, they're very high bar for treatment, especially in the checkpoint inhibitors, non-responsive disease. And so we really think that CAN-3110, when developed further, obviously, in clinical trial, will provide a very interesting alternative for the treatment of melanoma patients that are resistant to checkpoint inhibitors.
Yeah, I just want to ask you one other thing you mentioned earlier about immunotherapy and sort of the new frontier that is going on within the scientific world. This is an exciting time, I would think, in research for especially things like melanoma because of these things that we've seen over the last 5 to 10 years. This is a really good part to be in right now, isn't it?
It is. I think we are learning very much that we cannot treat all the patients with the same treatment. Obviously, when we started to develop checkpoint inhibitors, everybody thought that anti-PD-1 was going to be the answer for everybody. And now we understand that combination therapy is actually key because patients that are treated with checkpoint inhibitors and in general with immunotherapy tend to have a sort of a ceiling of response. And sometimes they need to drop these medications because they lack response, they developed resistance, or because of the side effects. And really with the viral immunotherapy, and we don't have only 3110, we are working also on other agents, it's really that type of population, these patients that do not respond to checkpoint inhibitors, for which we can really offer an alternative. And the other advantage, obviously, of using viral immunotherapy in combination is that the safety profile of these agents is extremely well tolerated and is described. As I mentioned before, we have data in recurrent high-grade glioma where this medicine has been directly injected into the brain in the tumors and has been well tolerated for the patient. Very similar data we have in the animal models that we're presenting here at SITC, where the animals tolerated extremely well the injections with 3110.
All right. Where do we go from here? What's sort of the next steps that people should be aware of as far as this one is concerned?
Well, we are excited about the possibility of expanding the indication for this agent. The melanoma is one of the space where we can leverage this drug, but there are others. So for example, triple negative breast cancer is a very high in expression of nestin. Sarcoma is extremely high in expression of nestin. So at the moment, we are undergoing a prioritization of potential avenues where to test these in vivo. Obviously, we are excited about the possibility of bringing this to patients as soon as possible. And we are collecting all the preclinical data, safety, tolerability, biodistribution, shedding that are necessary for doing these steps.
Yeah, incredibly important work for sure. Dr. Barone, thank you so much for updating us, and good luck with the conference, okay?
Thank you so much. Thanks. Have a nice day.
You as well. Dr. Francesca Barone, she's the Chief Scientific Officer for Candel Therapeutics.